Okay, can you see my presentation and can you hear me well?
Yes.
Okay, perfect. So hello everyone. As you heard, my name is Natasha Skoko and I'm heading Biotechnology Development Unit at the ICGEB in Trieste, Italy. And in this half an hour, I will try to share with you our successful story on the development of biologics and transfer of know-how to industry. Okay, let me just see you. Okay, so probably you don't need, I will just show you a few more, 2 slides about and a few words about International Center for Genetic Engineering and Biotechnology. You probably already heard from Emanuela in the previous presentations that we are intergovernmental, international independent organization with more than 80 signatory countries all around the world and 66 member states, and with the 3 main components in Trieste, Italy, New Delhi, India, Cape Town, South Africa. So we are an organization that focus our workflow and our interest in research, training and technology transfer to promote the sustainable development goals. So just quickly, as probably Emanuele already told you, that these ICGEB instruments of action are going from the research in our own labs in Trieste, New Delhi, Cape Town, through the advanced education supporting long and short-term fellowships for PhD students and postdocs in our member states. We organize different meeting courses, workshops at the international level, and we also provide grants for scientists in our member states, and also offer technical assistance in the regulation of biotechnology and its product. And last but not least, we worked in on the technology transfer to industry or in the field of the production of biotherapeutics and diagnostics. And this is exactly where my group comes, with more than 25 years ago, we started to develop expertise in the field of production of biotherapeutics and biosimilars. So maybe I can say just a few words. As we know, biologics or biopharmaceuticals are medicine produced from living organisms using biotechnology techniques. And we all know that they have revolutionized the treatment of anemia, diabetes, autoimmune disease, cancers in the last decades. And they are one of the fastest growing segments of the pharmaceutical industry. One biosimilar is a biological that contains a version of the active substance of an already authorized original biological product, so-called reference product, following loss of patent protection. So why similar? Because the similarity to this reference product has to be in the terms of quality characteristics such as biological activity, safety, and efficacy, that is based on the extensive comprehensive comparability exercise. So when we talk about biosimilars, we have to go back, as you can see on this graph from Data Monitor. Like in before 2000, the first generation of biosimilars came to the market. So we are talking about interferons, GCSF, so-called filgrastim, erythropoietins, insulins, growth hormone. And then on the stage comes the second generation of biosimilars that are monoclonal antibodies. They're fragments, proteins generated by fusion of antibodies and receptor moieties. So we are really proud to say that in Biotechnology Development Unit over more than 25 years, we worked on the development of the production of all these recombinant proteins that you can see circled in blue. So we really covered all most of the proteins that are in this first group, the first generation of biosimilars. So in the current wave, biosimilars have been limited to the inflammatory disease, oncology, diabetes, but as we can see now, and if we look all these new graphs that are coming now in the next wave, we will see biosimilars enter different therapy areas such as neurology, respiratory disease, ophthalmology, and rare disease. So we really think that the biosimilar opportunity is huge because biosimilars offer 35 to 65% savings to the health systems, and this is very important, and where their ICGEB mission comes on the stage, because the trend in the price reduction with the launch of biosimilars really resulted in the increase in usage. As you can see here, that a reduction in price up to 50 euro, as I said, in some countries up to 65%. It also comes together with increase in the volume of treatment per day. So this increase in usage means that more patients can be treated, that this increase in usage was heavily driven by this availability, of course, of biosimilars, but as well as other factors like expanded indications. That means that the doctors are more relaxed to prescribe this medicine also for different pathologies, because now they're cheaper and they are more available. So ICGEB came out with this model for transfer of know-how and to be somehow connected to the industry. Our mission was to strengthen capacity and competence of local partners to help our member states to accelerate the product availability at the more affordable price in our member states. So we really try to fulfill this mission, and we were thinking that the developing in-house expertise to produce these active pharmaceutical ingredients, to produce biologics, biosimilars is a good way. And also once we develop this expertise in-house, to transfer this know-how with no exclusivity, no royalties, really work on this unique model of the knowledge sharing. So I wanted just to address here that the technology transfer is a systematic transfer of know-how, knowledge, capability, experience, and documentation of product, process, or methodology between development and manufacturing. So it's the result of a partnership between academic and industrial fields, whose, of course, as I said, the main purpose is to make technology accessible to everyone, so technology transfer was really recognized as a key component of social economical development. It can accelerate acquisition of knowledge, of expertise in this pharma biotech field, and this is really necessary to build a strong local pharma sector. Through the technology transfer, of course, the decrease, it can result in the decrease of reliance of imports, raise quality, raise competence of local workforce, and accelerate the product availability at a more affordable price. So if you look at the biosimilars process development, we can see that it is almost 10 years long process, starting from the product development and comparative analysis, going through the process development, scale up, non-clinical, clinical studies, and finally to the regulatory agency reviews and approvals. So if you look in this pyramid, you can see that the biosimilars development it's really, it has on a base extensive analytical test, extensive quality control testing and comparative analysis between the biosimilar and the reference product. And this is the base for the development of biosimilar, but also that can extensively reduce the need for following preclinical and clinical data. So that's also one of the reasons why development of biosimilar and biosimilars by itself, they cost less than the original drug. So when we were thinking, okay, how shall what we need to develop in-house and in order to transfer that know-how to our member states, we thought that having the in-house this product development facility that can save every single partner and a guest that we work for at least a few years work. So that means that they can really speed up their process to the regulatory agency and final approval on their markets. So we think that we call it like technology in a briefcase. And if we look at this Ishikawa diagram of the cause and the effect, we really thought that what are every single step that you have to develop in-house. And of course, we had first to do what is the starting material and equipment and how to build the facility in order to develop cell line to produce the biologics, biosimilars, to have upstream process and downstream process expertise, and to be able to do on our own all the analytical methods, quality control tests, following European Pharmacopoeia monographs, and then offer it as an in-house training, write a good training protocols and report, and of course then all this technology in a briefcase could result in a successful technology transfer. So over 25 years, Biotechnology Development Unit at ICGEB build expertise in upstream and downstream processing. We have developed the different processes using all three platforms, bacterial, yeast, and mammalian cell platform to produce biologics. We have fermentation facility up to 30-liter bioreactor for microbes and 2-liter bioreactor for mammalian cells. We build the protein refolding capacity up to 50 liters and gain expertise in all kind of chromatography for the from the lab to pilot scale. So we are able to from some of these processes to purify and to have multi-gram scale batches of production. As many of these molecules are chemically modified, such as pegylated or lipidated, we have developed in-house a very good synthetic organic chemistry department that are able to do not just peptide and protein chemistry, to prepare all these polymers and do the conjugation with these chemical moieties and offer more molecules like pegylated interferons, pegylated GCSF, grow erythropoietin, and so on. And then, of course, at the end comes the very good quality control laboratory, because we have to show that the starting material, this concentrated recombinant protein, is really a good quality, so it can be a good basis to make a good biosimilar. So we have built, we bought all the equipment necessary to perform this dozen of the quality control tests that are written in European Pharmacopoeia monographs, with the Rever phase, capillary zone electrophoresis, glycan profiling, endotoxin residual DNA, mass spectrometry, and so on. So once we build this, then we thought, okay, that most of the industrial partners, they will not be happy if we offer them something like this. This is a small chromatography pre-packed column to purify the micrograms of protein. But our goal was to go to be closer to the industrial environment. So over the 20 years, we also developed some of these processes into so-called pilot scale. So we are now able to purify a few grams of proteins in one single batch, and we are using these big columns that are like having few liters of resin in order to purify like 30,000 doses of, for example, interferon alpha in one single batch. So once we develop all of this, I made a summary of what are the products that we have worked and we developed over the 25 years. So in bacterial platform, we have GCSF, pegylated GCSF, interferon alpha, pegylated interferon alpha, interferon beta. In a yeast production system, we have developed the technologies to produce insulins, long-lasting insulins, growth hormone. Any mammalian cell platform, erythropoietin and pegylated erythropoietin. So how does it work? So we have different steps in technology transfer and training model, and it's very fast and it's very straightforward. So we have we are in contact with the guests, with the companies from our member states. We then finalize the technology transfer agreement, and we offer from 4 to 6 weeks in-house training for the production of some of these biologics. So during this training, this is really hands-on training. So we repeat at least 1, 2, or even 3 batches of the production, purification, and quality controls in our own lab. We provide the protocols for downstream, upstream, and quality control procedures. And then if it's necessary, we assist also industrial partners in establishing the same process in their own facility. So we are really happy to say that in this more than 25 years of experience in the field of biologics and biosimilars, we signed more than 70 agreements with companies from 22 member countries. And we had more than 100 scientists trained in our own lab. In green, you can see countries where technologies have been transferred. And in green, you can see countries where these technologies reached market. And due to the pandemic, of course, in-house training during the 2020 and now in 2021, it seems that we were not able to provide in-house training. So we have developed an online training modules which can deliver training effectively everywhere. And I have to say that this helped us to sign the 3 contracts with the companies from Iran, from South Africa, and from Bangladesh to train them through the video-based training modules. So then all these years, we really suffered not having these pharma-like conditions in the lab, because we really had to move to 3 different buildings in the ICGEB in order to provide all the process flow. So this was really discontinuous process flow. And we really wanted to upgrade this lab and have the lab and lab equipment in a pharma-like conditions. So in order to go to the 2nd generation of biosimilars, to be able to produce monoclonal antibodies, and we really wanted, and we started to work on the trastuzumab and shetronlimab pegol, that are monoclonal, that are antibodies used to treat the breast cancer or rheumatoid arthritis. We really had an idea that to be closer to these industrial partners, we would really need the better equipment. We need the pharma-like, we need the clean rooms, pharma-like conditions in the labs. And we really would like to develop this master cell banking and the quality control lab for biologics and biosimilars under good manufacturing practice conditions. So this was our dream, to build a new facility and to offer and to have the clean rooms where we can do all the mammalian cell work, microbial cell work, and all the common processes as a downstream chemical lab and quality control labs. So our dream came through 3 years ago. We started this search for donors, and when you are searching for donors, of course, we wanted to convince them that we really, did a great job. We signed more than 70 contracts, and we have so many connections in our member states. And we really know what the company needs and what the industry needs. And we just need to build this new facility and to try to develop monoclonal antibodies production processes. And ideally, our 2nd stage of a search for donors would be that we found the donors who could cover the costs of training, so that our lab, our biotechnology development unit at the ICGEB, can in turn transfer this technology to the interested local manufacturers at no cost. Of course, we are self-financed laboratory. We are a non-profit organization, but we have to cover our cost of the development and running costs. So we have a small fee to ask for this company. So ideal situation would be really to follow our mission, that after the building the new laboratory, we would search and we need to find some donors that would cover cost of this training, so we can just focus on the training and transferring technology. So as I told you, we found the donor for the first part of our dream. That was the region of Friuli Venezia Giulia here in Italy, where Trieste is based. They provide us 3 million euro grants to build this laboratory and to buy and make this fully equipped laboratory, comprising the following spaces, like mammalian cell line development facility, upstream process, downstream process, polymer preparation, and the protein polymer conjugation lab, and the quality control lab in the GMP conditions. So we had 215, they gave us ICGEB gave us 215 square meters to build clean rooms. So we built the Class D that you can see here in green, clean rooms, and the Class C in orange, clean rooms for mammalian cell line development facility. With fully equipped, we bought the new machines like the clone picks, clone picker, imager, small modular bioreactors for mammalian cell like Amber 250 milliliter. We bought 2-liter twin bioreactors. We have 30-liter bioreactor, ACTA purifiers, ACTA Pure 25, mass spec, and so on. So this is our new lab. And I'm really happy to say that we have started building this lab like on the 16th of March last year. And during all this COVID pandemic, we worked really hard. And even if we had some interruptions in the construction place, we were able to finish on the 1st of March this lab. And if you allow me, I will share like 3 minutes video with you how our dream started, how the space looked like before and after 11 years hard work. So enjoy. Okay, I hope you enjoyed. And okay, and that would be half an hour, I think. I use all my time. And I just wanted to thank also, okay, this is my lab video team, and we are very small, but, dedicated people to this work. And also you can find all the information about our training and facility and the possibility of technology transfer and products on our new website, that is icgebb.com. And I would like to thank you for your attention.
Thank you so much, Natasha. And yeah, one of the things I think that is catchy from your talk is dreams do come true. If you don't give up, keep working at it. Okay, so we'll open the floor for anyone who has questions for Natasha, comments or conversation points. So Adelaide says, beautiful development and congratulations in the chat.
Thank you.
I think that in a way, Natasha, did we have transfers for African countries? Not yet, right?
No, we are now in the middle of transfer to the South Africa. Yes, we have an institute from South Africa, and maybe I can share with you. We have developed this video-based training now, and really we thought, oh my God, maybe people are not so happy not to have in-house training, but it seems that this was really good idea because everyone felt so disconnected 2020 last year that they really need any kind of connection or technical assistance and so on. So we have signed a contract with the institute in the South Africa, and now we are in the middle of that training. So maybe I can show you. It's very interesting. If you want, like, if you allow me, I can share with you how our video-based trainings now look like. But of course, we after this year, after pandemics, we hope to have these guests in our own lab, in our new facility. So I can just share with you. So on the website, like, we have video-based training, and let me show you just, if you allow me to go on our new website, I can share that with you. Okay, so let me share the screen. Where is the screen? I can't see now. Okay, so this is our new website, and this you can find all the products, technology, services, and so on. And if we go here on the video-based training, we have here got 3 companies that now doing the training, and one of them, so just to show you, so we have the training in the insulin production, for example, and we have developed really hard last year to work on the voiceovers, to put all the you know, the format as it should be for the video-based training. And then we have online assistance with them. So it looks like this. Let me show you.
Cell bank are used to prepare with 67 microliters of the working cell bank of Pichia pastoris GS115 IP strain.
That was just to give you a flavor how our online training looks like. But of course, you know, we would really like to have people in house.
So everybody can connect, right? And maybe what you can, so I can put the ICGEB.bdu in the chat.
Yes.
So if people are interested, this is the new, is this correct, Natasha, that I just put in the chat?
Yes, perfect, yes, yes.
Yeah. So this is the new address.
Yes, this is the new address. You find all the information, but of course, be free to write to me if you need any information about any product, the scale, or so we simply start talking to the companies, institutes, or whoever research groups. Just we've signed first non-disclosure agreement, and then we can freely start to talk.
This is really awesome, and I'm pretty sure a lot of people would be able to benefit from this access. So I would make a note of this website and possibly include that to the post-meeting email I will send out to people so that they can connect and, you know, get resources. But yeah, more questions, please, for Natasha, more conversation points. And I think I really like the fact, you know, I was just thinking that we've actually been able to reimagine what scientific training could look like. So you could be anywhere in the world and be able to benefit from exposure to, you know, scientific resources and training just by having a virtual means. And I think that's really cool and exciting. So yeah.
Yeah, and if you think, you know, we're really unique, you know, there are many research laboratories and everything, but we really follow the need of the industrial partners in our member states. So, and we are non-profit, you know, intergovernmental organization. We are not private pharma company. So this is something that, you know, our mission is not to have, you know, royalty or sign, whatever. It is just knowledge sharing.
Yeah. And I can see that Adrian has put a question in the chat. Said hi. It appears that your grants are only for academic-led projects, since the eligibility criteria states should hold positions at universities. Are you considering opening that to the open community following successful models like, for instance, Juggle or MIT Media Lab? It seems your mission statement is not limited to only academia.
Yes, yeah. So it's for sure not limited just to academia, because we, you know, ICGEB and Emanuele maybe can tell you even better. It was conceived with the idea to help industry. So it was UNIDA special project. It was United Nations Industrial Development, you know, organization special project. And this was really the mission from the start. Let's build something that we have academic projects, we have connection with academy and everything, but really follow the need of the industry.
No, definitely.
Thank you very much, Natasha. It's a very nice presentation and very nice information. I just want to ask you one question about the academic visitors in your institute. Do you accept application from academic visitor, research visitor in your section? What are the basic criteria for them, and what type of facilities you will provide it to them?
Okay, so how it usually works, we start first to talk and to see, you know, it would be good to have something in-house. So you need at least any kind of a lab and equipment, even in the institutions, in the research centers. So we worked a lot with the national institutes in different countries. You know, so even sometimes they want to have small laboratories, like the small biotechnology development unit. So what we do, we also clone these units in the different research institutions. So, and then, you know, that can be your help, then you can help building the biotechnology and transfer biotechnology to the industry in your own country. So we really, there is no limit. We work really with anyone. But of course, you know, in order to train people, you know, it would be good, and the transfer, the technology, it would be good that the research lab has at least, you know, equipment necessary to reproduce that process. So what we usually do, we train this, as I told you, all through the videos or between in-house training. And then ideally would be that once the guests go home, they can reproduce immediately the process in their own labs. So what we do, we prepare them before going, before coming to learn and to be trained. So we go through the equipment list, we see what do they have, what they need to buy, and how to, you know, which kind of resins, columns, and machines, and everything. And then once they're ready to receive the technology, then we go through the training process. So really, there is not, we worked on so many different models. You know, we worked with the big established pharma companies in different countries, like in South Africa, in China, in Russia. But also we work with the small research institutions, national laboratories. We worked in Syria, we worked in Turkey, in Uruguay, in Cuba. So really, we can adapt, you know, our model to your needs.
Yeah, so also, I mean, the good thing about is that we're very flexible. I mean, Natasha's unit. So sometimes they're just being, for example, making a protein or an enzyme for other institutes. So it's not like you have to have a strategic plan or if you need to make an enzyme, let's say, they have often done this. So
yeah, we have many collaborations in the institute and outside. Now, during the COVID, we were producing spike protein for and also some antibody to recognize the spike protein of SARS-CoV-2. So we have different collaborations, but we also can teach you and make a small trainings just in the part of the process. So maybe your own lab, needs some expertise in purification of the proteins, in different chromatography. So you can be just trained in that part. Or if we are interested in some quality control tests or some biological assay for some protein, we can develop with you or train you.
My lab has been using Natasha in the past few months to make a protein that is important for neurodegeneration, for example. When you need bigger companies, then not the BDU can also do this for you.
Yeah,
that's perfect. And another thing, if we don't have facilities in our home country, and I design some sort of project, for example, I'm working on THC production using immobilized strain. So I didn't have some sort of facilities in my home country. So I want to perform this project in MTL college, and similarly, I have opportunity here. Is there any opportunity in your lab from outside researcher who want to come and screen their project, work on their project to use your facilities?
So yeah, there are different like research grants that we can have, also different collaborations. So we just need to, as I told you, we are self-finance lab, so we have to find the space for different collaborations. We do have collaborations through the CRP projects and through the ICGEB grant programs. So we do have, but of course we have a limited time to dedicate because we are 8-person lab, so we can discuss, can help you and see which part of the project can be done. So we can have collaborations on short-term collaborations, like if you need just to spend 3 months in our own lab to finish your project. So that could be research collaboration projects.
And the technology transfer project, what is the basic need for the institution to apply for it?
Basic need,
basic requirement. For example, when I want to apply for this technology transfer, so what should be a basic need? What should be I design? I initially submit a proposal from this implementation, and what sorts of proposal I need, and what sorts of basic facilities I need to technology transfer. For example, recently I'm working on a development of my laboratory and in my home country. Basically, I'm working as a assistant professor in University of Turbat in Pakistan. So I'm going to establish a small lab for my research purposes, and this is basically a research purpose from similar, that is biotechnology based. I'm working on enzyme production. So I just want to establish in this lab. But basically, for example, a same lab can be used for insulin production if I try to do using same facilities.
So we have these 12 technologies that we offer. So these are ready to use packages. So this is our standard technology transfer scheme. So we have for every single step, we have standard operation procedures, we have a document, and it's really industry orientated. It's like the production process. So if you want to be training that, to give you just an idea, it could be possible, but as I told you, it would be nice that you have some facility where you can try it on your own. So we have all the list of the equipment, so we can be in touch. You can write to me and I can give you, if you're interested in the insulin process, I can share with you which kind of equipment it's necessary, what does it mean, how the training looks like, and everything. But it would be really good that you have something and where you can somewhere to repeat the whole process. So maybe you can be in contact with some of the pharma companies in your own country. So you can be then helping them to receive the technology and be a bridge between us or somehow,
So, for example, simply to add one thing, for example, for this COVID emergency, what the BDU did, they put online the whole procedure to make beta interferon. that can be downloaded by anyone and can be used eventually to make beta interferon. The idea is that the way that we like to work is so that people come here to learn, and then when they go back, they find an environment that is very similar, and this, of course, helps to transfer the know-how and the technology. If that is not possible, then there are probably a few other options, but that is the ideal way.
Thank you. Thank you so much for.
Okay, do we have any other question for Natasha? Speak now.
Thank you so much for your presentation.
Go for it.
Excuse me that hi, excuse me that I don't connect my video, but it's a bit of a mess here. I'm Daniel Guerra, I'm from Peru. Thank you very much for your talk, and very much in the line of Hanif, somehow like where to start, right? So maybe because we are in Peru here, the story from Argentina might be very instrumental for us to have an idea on what is the path to take. Do you have any document that more or less relates, narrates what happened? How was the development over there? How was the experience?
Experience, I'm sorry, I didn't get when the experience in Argentina, or what?
In Argentina. I'm thinking of Argentina because it's the most similar country we have in the map you showed.
Yes, we work with Argentina, yeah. Yeah, so usually we are approached by companies or research institution, and then we start to talk, to see what they could develop. We give them different advices, and then go through all the documents and see what would be the best place where they can try to reproduce that technology. So basically it's very straightforward. You just have to tell me what is your interest. You pick up the product, and we can start to talk, and then maybe you will get some ideas with whom to talk to. Argentina, we worked a lot with Argentina. We worked with a few companies in Argentina, and you know what, it's good because usually these big companies, they have a huge teams. So once you transfer them technology, because during the transfer we can train maximum 3 to 4 people, have 3 to 4 people in our own lab, but then that 3 to 4 people can train other 30 people in their own company, and this is like the start of their work. And usually it works like this: after the transfer they reproduce on the same scale, and then they need a few years time to scale it up, to adapt it to their own conditions. And it's really a knowledge sharing, because from that 3 people, that is like a primer, they build the teams of 30 or 50 people who then work and develop their in-house expertise to work on these biologics. And this is really our mission. Our mission is not to say, oh yes, I developed the perfect production process and it doesn't need any more optimization. No, our idea is I will save you 2 years time. You can have build your small team, your small biotechnology development unit, and then from there you can build your in-house expertise and work on that particular product that we train you, or even different ones, or even any product and any new molecules that you have in-house. Because once we train you how to use different chromatography, how to pack this in industrial columns, how to perform all this and read these European Pharmacopoeia monographs, then you have the knowledge, and you have an idea how to develop the further process on your own.
Yeah, thank you very much. Yes, the story you told is exactly what I was asking for. Thank you.
Yeah. Is there any more questions before I make some general announcements about the Reclone community? The options are going. In 30 seconds, be gone. Okay, so I assume there's no more questions. So thank you so much, Nat. Oh, Adrian, are you raising your hand?
Oh no, I said it's gone. No.
Okay, thank you so much, Natasha, for the wonderful, insightful talk, and also thank you for sharing with us the opportunities of resources and trainings and just where to start, you know, with this idea of knowledge transfer and setting up. I actually picked an idea of being a bridge between a company like a pharmaceutical and ICGEB, and you know, can start conversations there. So thank you so much for that. Okay, so as far as Reclone is concerned, I just wanted to make an early announcement that we are anticipating our next symposium to be in the first week of July. So you can get ready and start getting excited about that. The other thing is we would really appreciate anyone, maybe students who work with you and so on, who will be interested in volunteering to please step forward and support Reclone. There are a number of things that people could do. Help us manage the social media accounts and also help us design flyers, etc., which is really, I think, good experience for early career scientists or students or anyone actually to get involved in making this community really vibrant. So that is the information that I just wanted to share. So with that, I would thank everyone for being here, and just to remind you that this was recorded, so it's going to be uploaded on the YouTube channel in a few days. So if you want to go back and look through some of the resources that Natasha shared, and maybe you were able to take all the notes you wanted to, you can always have the video again. So thank you so much, everyone. Thank you, Natasha. Thank you, Emmanuel. Thank you,
Deborah.
everyone.
Okay.
All right. So
Okay.
have a lovely rest of your evening or whatever time it is, wherever you are.
Thanks.
Okay.
Thank you very much.
Yeah. Okay. Bye,
Thank
everyone.
you. Bye bye. See you.
Bye.
Bye. Bye.
Bye.